Hello and welcome to our experiment today on PhysioEx, exercise 8, chemical and physical processes of digestion. We have finished with activity 1 and 2, and now it's time to do activity 3, and that is assessing pepsin's digestion of proteins. So we have learned in the past that these enzymes need an optimal pH in order to operate. That's their properties. They need an optimal pH. They need an optimal temperature. If the temperature hyperthermia is going to denature the building blocks, these amino acids that make up the enzyme protein. And then if it's hypothermia, if it's too cold, it's going to freeze up and destroy this enzyme. So we learned about the properties of the enzymes. Now we're going to be learning about their specificity. We talked in the previous experiment that amylase is an enzyme that's specific for carbohydrates. It breaks down starch. And today we're going to be learning about how pepsin happens to be an enzyme that breaks down. It is a peptidase that breaks down proteins. So these proteases and peptidases break down proteins. And later on, we're going to be learning about lipase, which breaks down lipids. So the point is this enzyme specificity means that you cannot have lipase that digests carbohydrates also able to digest proteins and lipids. Does it make sense? And lipase that breaks down lipids is not going to be able to break down carbohydrates and proteins. And peptidases and proteases that break down proteins will never be able to break down these carbohydrates and lipids. So that's what we're going to be examining today. So we're going to be explaining how the enzyme activity of pepsin can be assessed with this very important assay known as BAPNA. I'll talk more about that in a little bit. And we're going to identify the substrate specificity of pepsin, meaning we're going to be targeting proteins. We're going to be discussing the optimal properties of the enzymes. So the enzyme needs the proper temperature, and the proper temperature is usually homeostatic temperature standardized for most of the enzymes, but the pH varies. So we have pepsin that needs really low pH because it works in the stomach, so pH 2. And you have lipase that needs, for example, pH around 7. So let's learn more. Okay, so in this activity, we'll explore the digestion of the protein that is made up of peptides or polypeptides. Peptides are 2 or more amino acids up to a number of 100. So peptide chain contains from 10 to 100 amino acids and is known as polypeptide. However, proteins can consist of large peptide chain about more than 100 amino acids or even multiple peptide chains. During digestion, chief cells they release this protein digesting enzyme known as pepsin. It's very interesting that chief cells begin with a C and they secrete P. So chief cells secrete pepsin, while parietal cells secrete with a C hydrochloric acid, HCl. So many students confuse, they assume that chief cells secrete HCl because well, they both have a C. No, that's not true. Chief cells secrete pepsin, and they assume that parietal cells beginning with a P, they also secrete pepsin. No, parietal cells secrete C, HCl, hydrochloric acid. So pepsin hydrolyzes these peptide bonds. This activity breaks up the ingested proteins and polypeptides into smaller peptide chains and free amino acids. So in this activity, we will use the BPNA as a substrate. So that will represent our substrate that will be taken into the active pocket, the active site of our enzyme of interest, pepsin. So BAPNA represents a polypeptide, and then we're going to assess its activity. We're going to see if we change the pH, will pepsin be able to hydrolyze BAPNA or not? And also we're going to be looking at BPNA. What is its structure? It is a synthetic peptide that releases a yellow dye product when hydrolyzed. So BAPNA is very helpful because we can tell. We don't need, like in the past experiments, IKI and Benedict reagent to tell us if the reaction went into completion. So all we need is that if the color of BAPNA changes into yellowish dye, you know that the reaction went into completion and the BAPNA reactant has been hydrolyzed and broken down into its reducing monomers, reducing amino acids, and now they are the product. So BAPNA solution turns yellow in the presence of an active peptidase, such as pepsin, but otherwise remains colorless. So if the solution remains colorless, BAPNA is intact and has not been hydrolyzed. So to quantify the pepsin activity in each test solution, we will use a spectrophotometer. So that's how we're going to be looking at the quantity, how much BAPNA has been hydrolyzed. And we're going to be using that to measure how much yellow dye is produced. So the spectrophotometer shines light through the sample of interest, and then it's going to measure how much light is absorbed. So obviously, the more BAPNA hydrolyzed, the more yellowish the solution, indicating more products being generated. That will translate to more light absorbed in the solution and less light transmitted and passing through the solution. So the fraction of the light absorbed is expressed as the sample's optical density. So yellow solutions where VAPNA has been hydrolyzed will have optical density is greater than 0. So the more BAPNA hydrolyte, the more yellowish the solution, the less light passing through, the more light absorbed, and the higher the optical density. The more subject, the more substance that's in the solution interfering with the passage of light from the spectrophotometer, that will represent higher optical density. So the greater the optical density, the more hydrolysis has occurred. So colorless solutions, in contrast, do not absorb light. So they have a very low optical density because there's no substance in there. They are clear. So all the light will transmit and pass through, and there will be zero absorbance, and will have an optical density near 0. So you need to understand optical density. So the higher the optical density, that means the more product we have, the more substance in the solution, they will impede and they will interfere with the passage of light that is going through the that's coming from the spectrophotometer, and then less light will transmit and more light will be absorbed. More light will be impeded, indicating high optical density. But low optical density, meaning that a lot of light is passing through unimpeded, uninterrupted. So some negative controls are included in this activity. With a negative control, a negative result is expected. So, for example, I know that if I have a clear solution and I pass light, I expect that, so for example, if I have a solution and I have a very, very high optically dense solution, I should expect no light transmission, and I should expect complete absorption and no light transmission. That represents an example of a negative result, meaning that I have no light passing through. Okay, so now let's get into the experiment. Where in the body does the protein digestion begin? So obviously the protein digestion begins in the stomach. That is where pepsin is being secreted. And the substrate for pepsin are proteins and peptides, both. In this activity, the substrate you'll be using to detect protein digestion is obviously BAPNA. That's the substrate. The substrate we will be to detect protein digestion is BAPNA. And negative results with negative controls are expected, and they validate our experiment. A spectrophotometer measures the optical density. So optical density is the opposite of transmission. So if light transmits, there's high light transmission. That shows there is low optical density, and that is shows there's high transmission. But if there is a low transmission of light, that means there is very high optical density blocking, impeding the passage or and the transmission of light. Okay, so drag a test tube to the first holder, and then the others are going to follow. So 5 more test tubes will automatically be placed in the incubation unit. That's our incubator. When we incubate, we're going to homogenize and mix all of the contents together, and we're going to make sure that everything is evenly mixed. So, and then the next step is let's now prepare the solution, and while we are preparing them, let's hypothesize. So tube one will have pepsin, so that's my enzyme, and it will have BPNA, and it will have pH buffer too. That's approximately the pH in the stomach. I mean, this is the perfect environment for the operation of pepsin. I mean, you have the pepsin, the enzyme, you have its substrate, and you have its optimal pH and the temperature 37 degrees Celsius. I mean, this is perfect. And so we expect complete reaction here. Let's look at two. You have pepsin, that's my enzyme. You have BPNA, that is my substrate, and the pH buffer 2. So that is the same thing as tube 1. So that is that we should expect 100% reaction. Now let's look at 3. 3 says we must have pepsin, that's my enzyme, and deionized water and pH 2. Well, that's interesting. I have my enzyme, I have the proper temperature, 37 degrees Celsius, and I have the proper pH, but I have no substrate. So I will not have a reaction. So it's never going to turn yellow here. So tube 4. Tube 4, you're going to have deionized water, BAPNA, BAPNA, and then you're going to have pH 2. You're going to have the substrate and the proper pH and the proper temperature, but no enzyme to catalyze the reaction. So it's never going to turn yellow. Then you're going to have pepsin, that's my enzyme, and you're going to have a BAPNA, and that is my substrate. My temperature is optimal, but my pH is 7. That approximates the pH of the mouth. That is not the proper pH of the stomach. So now I know that the reaction is not going to be going to completion as well as it will in the stomach pH too. So I should see the yellow color very light. I would not have bright yellow because the enzyme needs its properties optimally need optimal properties fulfilled in order to do its job. Here, the same thing. So I have pepsin, I have my enzyme, I have my product, and I have my buffer 9 that approximates the pH at the intestine. So I don't, the same thing, I don't expect it because I have interfered with my proper pH for my pepsin, that's 2. So I do not, I give pepsin 37 degrees Celsius, but not the 2 pH. So now we're incubating, and this represents, 1 hour, but it's going really fast. So now let's see. Okay, so yellow, light yellow, really weak yellow. And now let's now perform the spectrophotometer. We were supposed to boil them. We were supposed to was to incubate. Then after we prepare, then we're supposed to boil them. And then after that, as a question, which of the following two are negative control? Negative control meaning I should get nothing. And I can tell, deionized water. And so I should have my enzyme pepsin, and that sounds like a negative control. So because I have my pepsin, and I have my water, and so 3 and 4 sound like negative controls. We incubated. Which page do you think Pepsin will have the highest activity? Obviously, too, because it's stomach. Question. Analyze the optical density. We recorded that. We're going to put it back. Take 2, analyze, record data, put it back. Take 3, analyze, record data. So I forgot to boil it, but it's just one of the steps. 3 and then 4, analyze, then put it back, record data, then 5, analyze. An optical density greater than 0 indicate that an optical density greater than 0 indicate that BPNA digestion has occurred definitely, and pepsin is active. That is definitely true. Okay. Okay. So let's examine our results after we submit. We submit. Okay. Pepsin would be most active in the stomach. That's true. Which 2 tubes validated the results of the experiment? These are the control tubes. So obviously 3 and 4. Pepsin, deionized water, pH 2. And so this one has a substrate and no enzyme. This one has an enzyme and no substrate. They validated the experiment. The more enzyme activity, the optical density, the more enzyme activity, so if the pepsin is, the more active pepsin is, the more products I'm going to have. And the more products I'm going to have, the more substance I'm going to have in my test tube blocking and impeding the transmission of light. And that will increase the optical density, make a really dense and thick solution. So that increased the optical density. If pepsin were digesting an actual protein substrate, the product would be peptides and amino acids. Because remember, peptides are from 10 amino acids up to 100. So you can have a huge globular protein that when you digest it, you break it down into not just peptides or not just amino acids. You may need additional rounds of enzymatic digestion to break down peptides into even more amino acids. So we don't just go from peptides into amino acids. We don't just go from polysaccharides into monosaccharides. We go into different routes. So sometimes you have polysaccharide plus monosaccharides. Sometimes polysaccharides, disaccharides, and monosaccharides. Sometimes polysaccharides, even more polysaccharides that are smaller than the main polysaccharide, in addition to the disaccharide, than the monosaccharide. So it's definitely peptides and amino acids. I really want us to look now at the results. So now let's look. So we have not boiled them, but we should have. We should have boiled them. But the point is that in experiment 1, I have pepsin and BPNA with the proper pH and a proper temperature. Then that shows me the proper active, that there is really high optical density. Everything else, so this tube 3, it has no substrate, so it's 0 optical density because the reaction never went to product. And here the reaction never went to product because I do have a substrate but no enzyme, so I got a 0. Here I have a subliminal reaction that happened. I do have my pepsin enzyme and I do have my substrate, but my pH is 7. So I have a little bit of reaction that happened. So that just, it's very interesting to show you that, for example, pepsin that works better at optimal pH of 2. If you increase the pH, the farther away you go from a 2, the less it's going to work. So now you can hypothesize if you run this experiment again with a pH of 3, 4, 5, 6, 7, 8, 9, 10, as you go upwardly in the pH. So pepsin will do better at pH 2. It'll do less better than at pH 3. It'll do even less than at pH 4. So the farther you go away from 2, the less the reacting going to go into completion. And the more you approach 2, the more the reactant are going to go into completion. Submit. Okay, describe the effect that boiling had on pepsin and how could tell that it had an effect. Well, unfortunately, we did not do the boiling, but what we should see is that boiling will denature the the enzyme. We talked about hyperthermia, denaturing the proteins and breaking the bonds between the primary to secondary to tertiary structure of the proteins. What was your prediction correct about the optimal pH for pepsin activity? At which pH do you think? Yes, that is true. And what do you think would happen if you reduce the incubation time to 30 minutes for tube 5? So if you make the reaction, so you have pepsin, BAPNA, and pH buffer. So if you reduce the time, you're going to have even less because the pH is not even optimal. It's 2. The optimal pH is 2. Now you went up to 7. That is about 5 more units away from the optimal pH. And now you're going to reduce the time even more. So the optical density is going to happen to be even less. And you're going to have less products and less reaction going into completion. Okay, let's see if we can just quickly, let's see if we can do the boiling one more time. But yeah, let's see if we can do the boiling. So to 1 pepsin. Okay, so now let's go do our boiling. Select the number 1 under the test tube. Okay. We boiled. So we're gonna boil all of them. Then we incubate them. Okay, I think that'll do it.